Hello you guys, in this video we are going to transition into the Unit 1.2 notes. In these notes we're going to dive in a lot deeper into macromolecules and the different types of macromolecules that are found in living things and what they do and the structures of those molecules. So in the last set of notes we ended with this talking about or doing kind of an intro to organic chemistry. And I told you guys that there are these very large molecules that we often find that in cells and living things that living things are made of, and we call them macromolecules. And so I'll do a quick refresher of a couple of things from those notes, and then we'll start going into topic one of these notes. And topic one of these notes, we're going to specifically look at a certain type of macromolecule called carbohydrates in topic one. And so there's four different types of macromolecules that basically most organic molecules fall into one of these categories that we find in living things. And so in this video, we're specifically going to focus on carbohydrates. And so just some quick background refresher, just to make sure you guys, this is fresh in your head. That living things were made of these organic molecules that have these carbon hydrogen backbones with functional groups attached to them, and a lot of times these molecules are very large and they form what we call macromolecules. And there's four classes of macromolecules that we're going to look at in the Unit 1.2 notes. It's carbohydrates, lipids, nucleic acids, and proteins. All of these are very important types of molecules that are essential for life. Now most of these macromolecules exist as polymers, which are basically these giant molecules, but really they're just made up of these repeating subunits called monomers. And so we see these certain molecules that are just repeating and binding to themselves over and over and over again. And that's what we're going to see a lot in these notes when we're looking at these macromolecules. Basically, they're just polymers made up of these monomers. And then just real quick, there's a huge diversity and variety of these polymers that exist in living things that are all built from a small set of monomers. So with these small building blocks, we can make a lot of different things. I kind of use the analogy of Legos. With just a small number of different types of Lego pieces, you can build a very large amount of structures that do different things. And so each cell on this planet contains thousands, tens of thousands, hundreds of thousands of different macromolecules that are performing a variety of super important functions. And ultimately, a key concept in these notes that we're going to be referencing a lot is that the function of these macromolecules and what these macromolecules do and their purpose in cells is going to be determined by their structures. Their structure and how these macromolecules are built and what their actual structure is going to determine and affect its function and ultimately its role in cells and living organisms. So let's get into carbohydrates. So carbohydrates, these are a class of macromolecules whose function, they kind of have two main functions or types of functions. One function that carbohydrates have is energy storage. So we use carbohydrates in cells and in living things. Carbohydrates are used to store energy, and we can tap into these carbohydrate molecules to get energy from them as our cells need that and are using that energy. And usually this is shorter term energy. So this is energy that we're storing and using in the same day or in a couple of weeks. It's not long-term energy storage, which we'll talk about later. But this is energy that we're storing in our bodies and using kind of on a regular basis. And that's one function of carbohydrates. And then another function of carbohydrates is they provide, they have a structural function in that there are some really important carbohydrates that build certain structures inside of cells and living organisms. And so they provide building material for cells and life on this planet. And inside their bodies and inside their cells, they're going to be building these very important structural components. So there's an energy storage function where certain carbohydrates are used for energy. And then there's a structural function where some of these carbohydrates are used to actually build different components of cells and living things. The elements that you're going to find in carbohydrates, which is something else that you're going to need to know and start memorizing. It's usually the smaller details that you guys struggle with. So you need to know the actual elements we find in carbohydrates, because later on we'll see how that's a little bit different than some of the other macromolecules. But in carbohydrates, you're going to find a bunch of carbon, a bunch of hydrogen, and a bunch of oxygen. And that's basically it. There's going to be carbon, hydrogen, and oxygen. And those elements, those types of atoms are what build carbohydrates. And so, like I said, carbohydrates, like basically most macromolecules, these are polymers, which are just these super massive molecules made up of repeating subunits called monomers. Now, specifically, when we're talking about carbohydrates, the monomers are called monosaccharides. So monosaccharides is the name of the monomer that builds carbohydrates. And this is what they look like. These are different monosaccharides. We also call them simple sugars. And here's some of their names, and there's other ones too. This is just a sample of different ones. They all kind of have this basic ring structure that you see in some of these guys right here. Here's glucose. You guys are probably most familiar with him. Glucose is a monosaccharide, and he's used to build carbohydrates. And so we'll see him a lot in these notes, actually. But then there's some other ones here, these simple ring structures. And they, in these monosaccharides, you're always going to have something very close to a 1 to 2 to 1 ratio between carbon, hydrogen, and oxygen atoms. So like glucose is C6H12O6. It's a 1 to 2 to 1 ratio. Another example is deoxyribose. This is the sugar that's actually found in your DNA, or used to help make DNA, which we'll talk about later in these notes. But the formula for that is C5H10O5, so another 1 to 2 to 1 ratio. Anyway, these are monosaccharides. Now, what's going to happen is these monosaccharides oftentimes are going to start binding to one another to start building these larger polymers. Now, it's worth noting that another common thing that we find is something called a disaccharide, and that's when two monosaccharides bond together. So these are also pretty common or popular. So I just want to point them out real quick. But a disaccharide is just two monosaccharides bonded together. And they're bonded together via a dehydration synthesis reaction. If you guys remember, in the last set of notes, we talked about dehydration synthesis reactions, which is the type of reaction that puts new bonds between monomers. So in this case, we're looking at monosaccharides, and there are dehydration synthesis reactions that put a new bond between them. That bond that connects monosaccharides together is called a glycosidic linkage, which is something you guys have to know. So this bond that's connecting a monosaccharide with another monosaccharide, or monosaccharides with other monosaccharides, we call those glycosidic linkages, which again are formed by dehydration synthesis reactions. And so there's some pretty common disaccharides that exist that you probably have encountered in real life and living so far on this planet. Sucrose is a disaccharide. It's made up of the monosaccharides glucose and fructose. So you take a glucose, you connect it with a fructose, and now you have what's called sucrose. Sucrose, this is table sugar. This is the sugar that you buy at a grocery store. You probably have in your kitchen somewhere. This is that. Lactose, this is the sugar that's found in dairy products. So in milk and cheese and yogurt, there's lactose, which is a sugar that's made up of two monosaccharides called galactose and glucose. And so that's what disaccharides are. And so again, just remember, and I will point this out multiple times throughout the Unit 1.2 notes, there's dehydration synthesis reactions, which are responsible for creating these new bonds and joining monosaccharides together. And then you could also do the opposite. You could take hydrolysis reactions, and you could break the monomers apart, which also is very common and happening a lot in your guys' cells. In terms of these monosaccharides being broken back into simple sugars. Just an interesting side note here, or a specific example, I talked about in the last notes these things called enzymes, and enzymes are these molecules, which we're going to talk a lot about later on in Unit 1. It's these molecules who are responsible for controlling these reactions, and they're very specific. So for example, lactose, the sugar found in milk that I pointed out right here, this sugar, when you guys are eating these disaccharides in your body, your body can then use these molecules and break them down into simpler sugars that can then be used for energy for your body as part of your metabolism. And lactose is one of those. And there's a specific enzyme who's responsible for hydrolyzing this. So a hydrolysis reaction breaks this apart. And there's a specific enzyme called lactase. And this enzyme is a special molecule who's responsible for allowing lactose to do a hydrolysis reaction and become lactose and glucose, these simple sugars. And so what's interesting is just to connect this to things that you might have heard about, like for example, it's very common for people to be lactose intolerant. If you're lactose intolerant, it's because your body is not creating a lot of this enzyme that's responsible for breaking down lactose into these digestible sugars. And so then when you consume lactose, this sugar, this disaccharide, your body and your cells and your digestive system is not very happy because it's not able to break it down into these simpler sugars that the body can then metabolize and use for energy. And that's because of this enzyme here. And so if you're not lactose intolerant, it's because you have plenty of lactase being produced in your body. What's also interesting is that everyone at one point was not lactose intolerant. When during your life, very early on when you were a baby, actually everyone was making this enzyme because you relied mainly on milk for when you were a baby, and in that milk there was a lot of lactose sugar, and this enzyme was used to break down that sugar. And what's actually supposed to happen is as you get older, the gene, there's actually you have a gene in your DNA that codes for this enzyme, lactase, and that gene is supposed to stop being used by your body, and you're supposed to stop making lactase. So really, people who are lactose intolerant, those are the normal people, because really you're supposed to be making this enzyme when you're younger, and when you're older, you're supposed to start not making this enzyme anymore in your body. But what happened thousands of years ago is there was a mutation that occurred that allowed people to continue making this enzyme into adulthood. And so now we have, it's very common for adults to still be able to consume milk products and this lactose sugar and be able to digest it, because they've inherited a mutation from a long time ago in human populations that existed a long time ago, who started becoming very agrarian and started farming and started having cattle that they would actually drink the milk of, and they relied on that for nutrients. And there was this mutation that occurred that was super beneficial that allowed them to continue making this enzyme as they got older. And so if you're not lactose intolerant, you're actually the mutant. You have a mutation that allows you to not be lactose intolerant. It's kind of interesting, but we'll talk more about that stuff later this year. Anyway, continuing on with this idea of carbohydrates, these monosaccharides, like I said, they join together and they can form disaccharides. But beyond that, really what's usually going on is these monosaccharides, these monomers, are going to bind to each other over and over and over and over and over again, building these massively huge polymers. Now, if we're talking about carbohydrates, the name of the polymer is a polysaccharide. So a polysaccharide is a polymer made up of monosaccharides. So when you have a bunch of monosaccharides bonded together, we call it a polysaccharide. So that's the polymer, the name of the polymer when we're talking about carbohydrates. So the monomer is monosaccharides, the polymer is polysaccharides, and it looks a lot like these molecules that I have pictured here, where it's just a bunch of monosaccharides, those simple sugars that I showed you, binded over and over and over and over and over again. And these are super massive molecules, these polymers that are forming. And so there's two main types of polysaccharides, or two main functions, kind of like what I said earlier for these polysaccharides. Some of these polysaccharides have an energy storage function, and their purpose in cells and in living things is to be a source of energy that can be used. And then there's some polysaccharides that have a structural function, and their purpose is not to be used for energy, but they actually build different important components of a living organism. And so let's start with storage polysaccharides. Storage polysaccharides, like I said, are used to store energy so that cells can then tap into these molecules for energy. And that's what I actually have pictured here. These are two storage polysaccharides. In plants have a storage polysaccharide called starch that they make. So starch is actually a polysaccharide. Looks like this, and it's actually made up of just a bunch of glucoses. So it's glucose, 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 bonded over and over and over again. And plants build this inside of their cells and store this inside of their plant tissue so that they can then use this sugar basically whenever they need to. They can tap into these sugar molecules and then use that to make ATP and to power their cells. Animals basically have something super synonymous. It's called glycogen. Glycogen is a storage polysaccharide found in animal cells. And this is very similar to starch. It's actually also made of glucose. It's just a bunch of glucose bonded over and over again, just like starch. They're both very helical structures, and they form like these helixes that you see here, these twirls. And the only big difference between starch and glycogen, other than starch is made in plants and glycogen is made in animals, is that glycogen has a little bit more branching. So there's a lot of more branching off on this molecule. And in starch, there's not as much branching going on. But basically, they're the same type of molecule. And so what happens in your body when your body needs, or when in an organism, when that organism needs energy, it can just chip away at these gigantic polysaccharides and start separating these glucose monomers into just free glucose, just glucose by itself, individual molecules of glucose, these monosaccharides, that can then be used in your cells. Your cells can easily use this to make ATP and to provide you with energy throughout the day. And so, and that would be through a hydrolysis reaction. Hydrolysis reactions can break these bonds when needed and give your body a bunch of sugar whenever it needs it throughout the day. So during the day, when you guys are eating a meal, or like when you eat a meal, like breakfast or lunch or something, what's happening is you're taking in all of this. A lot of times that food contains carbohydrates or sugars. And those sugars are going to be digested, and then in your body, those sugar molecules are actually going to be taken in by cells and used to build glycogen. Your body, after a meal, it's going to try to save as much of that sugar from your food as possible. And your muscle cells do this a lot, and your liver cells, and your muscle cells and liver cells will start building glycogen through dehydration synthesis reactions. And then that way throughout the day, because you're not eating constantly, but later that afternoon, it's been a few hours since you ate, you're running the mile for PE, or you're going to sports practice, and your body needs a lot more energy now, your muscles need a lot more energy. They had this saved, they stored it, and now they can start chipping away at it to deliver this sugar that they saved to your cells so that they can use it for energy. And so that's what glycogen is doing inside of animal cells, like our cells. We build glycogen. Starch is the same thing in plants. They build starch just to store energy. Now, structural polysaccharides, these are also polysaccharides. They're made up of monosaccharides bonded together over and over again. But in structural polysaccharides, their purpose is not to be used for energy. They actually are building components of cells or living things. So one example is like cellulose. Cellulose is found in plant cells, and it's used to build the cell walls of plant cells. You might have learned at some point that plant cells have a really strong, rigid cell wall that surrounds the cell. That's very sturdy. And provides a lot of structural support to plants. Animal cells don't have a cell wall. We'll talk about this later. But that cell wall in plants is made of cellulose, which is actually, again, a polymer, a polysaccharide made up of glucose, again. So glucose is very common. And so it's just a bunch of glucoses bonded together over and over and over again. Another example of a structural polysaccharide is chitin. Chitin is a polysaccharide that's used to actually build the exoskeleton of certain animal organisms, like insects, their exoskeleton, and in crabs and lobsters and crustaceans, their exoskeleton. We find this molecule. It's also built for fungi, like mushrooms and stuff like that. Their cells also have a cell wall, but their cell wall is made of chitin, and it's not made of cellulose. Anyway, these have a structural purpose. Their purpose is to build structural components inside the cell, but these are also polysaccharides. Now, ending these notes, I just want to talk about why do these polysaccharides have different functions? And specifically, a great example is like cellulose, which is just made of a bunch of glucoses bonded to each other, and I pointed out starch earlier, which is also made of a bunch of glucoses, and both of these are made in plant cells. So in plant cells, they're building starch, which is just a bunch of glucoses bonded together, but they're also building cellulose, which is just a bunch of glucoses bonded together. And what makes them different? Like, why does one have a function for storing energy and one have a function of building this structural component which is the cell wall in plant cells. Like, why is their function different? And their functions are different because the structure of these molecules are actually different, which I'll explain in a second. But the structure of cellulose is different than the structure of starch. And they actually are, even though they're both made of glucoses, their three-dimensional structure is a lot different actually. And so I'll show you that in a second. But basically it's because they're going to have different structures because the glucose monomers that they are using are actually a little bit different. There's actually two versions of glucose. They're called isomers. There's alpha glucose and there's beta glucose. I'll go ahead and show you this picture here. It's basically glucose. This is glucose, this is glucose. They just are arranged a little bit different. You guys don't have to really worry about this. Just know that there's two versions of glucose. Now, if you're putting the alpha glucoses together, the glycosidic linkages that connect them, these bonds, remember I told you these are called glycosidic linkages, these new bonds that are forming between them are arranged in a certain pattern that's actually going to be different than when beta glucoses are bonded together. So when beta glucoses are bonded together, the glycosidic linkages have a slightly different pattern. You can kind of see it's an up, down pattern. And because of that, because starch and cellulose, which are both using glucose, are using different versions of glucose and have a different arrangement of glycosidic linkages, those glycosidic linkages are arranged slightly different, that actually gives them two completely different three-dimensional structures. So in starch, the way that those glucoses are bonded together with those glycosidic linkages actually creates these helical structures. So starch is very helical. It winds up like this. And there's some branching that occurs a little bit. But it's very messy molecule, but it's very good at just being somewhere in a cell or in plant tissue, and then it can be tapped into for glucoses. Now cellulose, there's a big difference. In cellulose, because of those glycosidic linkages being a little bit different than in starch between these glucoses, it actually forms very linear, like super linear strands of glucoses, where these glucoses and their glycosidic linkages are arranging themselves in a way where they're not going to actually spiral. They're going to form these very straight linear strands. And those straight linear strands of glucose are very organized and can lay on top of one another and form hydrogen bonds. And so there's hydrogen bonding that's occurring between these glucoses and one of these strands and another strand and another strand, because it's a very organized molecule. And all of that creates a very strong, sturdy molecule. So cellulose is a very strong molecule that's perfect for creating a very strong component that's needed inside the cell, like the cell wall for plant cells. And so again, this difference, and you see there's no branching, so there's no branching, it's very linear, it's not helical, there's a lot of hydrogen bonding occurring between these parallel strands that form these bundles called microfibrils that are very, very sturdy, building the cell wall of plant cells. And it's because of these glycosidic linkages being arranged a little bit different between these glucose monomers and starch versus in cellulose, because they're using two different versions of glucose. Anyway, that's it for topic 1. All of this was about carbohydrates. So hopefully you guys know the basic idea of the different functions that carbohydrates have, whether it's storing energy or structural, building different components inside of cells. And you know a lot about their structure too, and how they're made up of these monosaccharides that bond together to form polysaccharides, and kind of what that looks like. Anyway, thank you guys. I'll see you guys in class or in the next video.